Prior to capturing cells on the BD Rhapsody single-cell analysis system, cell sorting may be required to enrich specific cellular phenotypes. In this tutorial, we will walk you through our recommendations for how to co-stain both oligo-conjugated AbSeq and/or sample tag antibodies with fluorochrome conjugated antibodies that share the same clone. In doing so, you can save time and sample with one antibody panel for your entire workflow from flow sorting to single-cell capture. If you want to follow along with our example, you can download the technical bulletin from our website. If there are no overlapping specificities between the fluorescent and oligo-conjugated antibodies, then you can proceed with your standard staining protocol or the one recommended in the technical bulletin. However, if using the same clone of the same specificity for both fluorescent and oligonucleotide conjugated versions, we'll need to determine the volume of both antibodies so equal microgram quantities of each are used. In our example today, we'll be using a single shared clone. For these two, when calculating equal microgram amounts, we will mix 8 microliters of flow antibody and 2 microliters of AbSeq antibody for a total of 10 microliters. The shared clones are stained first, so make sure to keep this pool separate from the other antibodies used. Our staining protocols have been validated for a final staining volume of 200 microliters, so keep that value in mind as we move forward. A flow proxy assay can be used to further titrate AbSeq antibodies if required. We will next calculate the volume of each additional antibody used in the assay that has a unique clone. In our example, we have 5 fluorescent antibodies and 10 AbSeq antibodies. We will be using 5 microliters each of 5 fluorescent antibodies and 2 microliters each of 10 AbSeq antibodies for a total of 45 microliters of non-overlapping clones. We add this value of non-overlapping clones to the previous total for the shared clones for a grand total of 55 microliters of antibodies. Pull the non-overlapping antibodies in a separate tube than the overlapping ones because they will be added later in the staining process. Next, we'll subtract this total volume of antibodies, 55 microliters, from our desired volume of 200 microliters to obtain the volume of BD stain buffer that will be used during staining. In our case, we will need 145 microliters of stain buffer. If your cells are not in the appropriate volume or buffer, pellet them and then resuspend the cells in BD stain buffer. In our example, we will add 145 microliters of BD stain buffer and the 10 microliter pool of the shared clones. Remember in our example, we had 8 microliters of flow antibodies and 2 microliters of BD AbSeq antibodies in our pool. Then, pipette mix and incubate the cells for 10 minutes on ice. After the first incubation, add the remaining 45 microliters of fluorescent and BD AbSeq antibodies to attain the final volume of 200 microliters. Pipette mix and incubate the mixture for 30 to 60 minutes on ice. After staining is completed, transfer the labeled cell suspension to a 5 milliliter polystyrene Falcon tube and add 2 milliliters of BD stain buffer to wash the cells. Pipette mix and centrifuge the tube at 400 Gs for 5 minutes or an appropriate speed and time for your cells. Decant the supernatant, keeping the tube inverted. Gently blot the inverted tube on a lint-free wipe to remove residual liquid from the rim of the tube. Do not tilt and decant the tube multiple times to get rid of fluid, as this may dislodge the cells from the tube. Repeat the washing steps 2 more times using 2 milliliters of BD stain buffer for a total of 3 washes. After the final wash, decant the supernatant as instructed earlier and resuspend the cell pellet using the appropriate volume of BD FACS pre-sort buffer, considering cell density and sorting conditions. Now, we're ready to proceed with our flow sorting workflow where specific cell phenotypes will be enriched or isolated based on the fluorescent antibodies used during staining. Post-sort, we can proceed with single-cell capture and cDNA synthesis using the BD Rhapsody single-cell analysis system. If you have any questions, additional recommendations are provided on our FAQ section at scomics.bd.com or reach out to our multiomics help desk. Thanks for watching.